So the ability to identify microbes is one of the most important parts of microbiology for healthcare professionals. Why? Because this is what helps us to diagnose and treat infections. So in chapter 15, we're going to be looking at diagnosing infections. And we're going to start by looking at phenotypic methods. There's going to be three different methods. All right, so here are those three different methods. We have phenotypic, all right? Phenotype is physical characteristics, okay? So we're going to be looking at the physical characteristics, the macroscopic and microscopic morphology, which means body structure, physiology, biochemistry, all right? So then we also will look at immunologic, okay? And this is going to be serological analysis. We're going to use antibodies or the patient's antibodies, prepackaged antigens, and look for those, okay? And then lastly, we'll look at genotypic, okay? The genotypic or the genetic techniques that we can use as a sole resource to actually identify bacteria, okay? Data from these methods can provide a very unique profile for any bacterium. So phenotypic, actually, I think we've gone through all of those. So genetic means of identification are often being used solely now as a resource for identifying bacteria. Increases in what we call the point of care diagnostic test can be performed, and samples don't have to be sent out, okay? And so the patient can continue on without being delayed. That's one of the benefits there, okay? These include CTs, MRIs, and PET scans. So in phenotypic, we're going to be looking at the observation of traits that the organism is expressing. What is part of their metabolism? What enzymes are going to be at play? Are they breaking down sugar with a particular enzyme? We can test for that, okay? What is their appearance? Are they cocci or are they bacillus? Are they gram-positive? Are they gram-negative? All right. What physical conditions can it live in? What does it thrive in? Is it aerobic or anaerobic? What antibiotics are it susceptible to? What is the composition of the cell wall, the cell membranes? Is it gram-positive or negative? All right. Then immunologic, the antibody response is going to be exploited for our diagnostic purposes whenever a patient's sample is tested for the presence of antibodies to a suspected pathogen. Okay. Often easier than testing for the microbe itself. You can have lab kits that are available to ID a number of pathogens immediately. All right. Genotypic, some of the advantages of genomic methods over phenotypic methods. You don't necessarily have to always culture the microorganism, okay? These are increasingly automated. They give us very quick results that are often more precise than phenotypic methods. We have what we call viable non-culturable microbes that cannot be grown in the lab. These are easier to be identified with a genotypic method. So in looking at phenotypic methods, what is one of the first steps? Well, that's going to be specimen collection, okay? Care should be taken with all samples that have resident microbiota to prevent any contamination. Only the infected site should be sampled, not the surrounding areas, and we must use aseptic techniques, okay? And so if you want to be successful, the way you handle it, collect it, store it, and culture it are all going to be important. Sterile containers are going to be used to prevent contamination of the environment or from the patient. Examples of samples here for collection, you can see here in this picture: saliva, sputum, swabbing the mouth, the nasopharynx, or the throat to reach the tonsils, blood, urine via clean catch methods or catheter, skin swab, spinal tap to get the cerebral spinal fluid, feces, vaginal secretions via swab or stick. Skin samples can be obtained with a scalpel. You can use a culturette to get components. So saliva, skin, and spinal fluid are all appropriate specimens to use for bacterial cultures. In all sites that have those normal microbiota, you need to be careful to only get the infected site and not that surrounding area. Saliva has millions of bacteria per milliliter that are normal microbiota. Sputum. Sputum is going to be the mucus secretion coating the lower respiratory surfaces in the lungs, that type of thing. So sputum is going to come out by coughing, or it can be removed via catheter. Contamination with saliva should be avoided because it does contain so many millions of bacteria that are normal microbiota, but it's going to make it very hard to test. The only truly 100% aseptic extraction of urine is going to be with a catheter. Okay, you can do a clean catch, and then you are going to wash the external genitalia, urethra, and such, and collect the urine during midstream, and so that's going to greatly reduce the number of external pathogens. Okay, external microbes. All right, so contaminants. All right, so any contaminated microbes from the clean catch method can generally be differentiated from the pathogens, because we're going to reduce that greatly. After proper collection, well, actually, let me back up here. Dirty catch is going to be the first avoided urine. Sometimes that is required for different diagnostic techniques. And then, like I said, you can also swab the vagina, urethra, or cervix. After proper collection, specimens must be transported to the lab and stored appropriately in a aseptic way. Okay, usually stored in a refrigerator. Non-sterile samples like urine, feces, and sputum deteriorate very rapidly at room temperature, so this is especially important for them. Other sources for specimens include ears, ear canal, eyes, synovial fluid, nasal cavity, all done via swab, diseased tissue surgically removed or biopsied. So labeling and identifying these specimens is very important, as well as providing accurate patient history, and this is crucial to obtaining timely and accurate results. Okay, you can also sample the blood, cerebrospinal fluid, tissue fluids by aspiration, where you take a needle and pull it out. Okay, but in those cases, it is very important to do antisepsis of the puncture site. So you've got to clean the skin before you run the needle through it. Right. So this is an old school form here showing various tests that can be requested. Most places now are electronic, but this is a good thing to kind of start at and look at. Patient analysis for signs of microbial infection should be monitored. Fever, wound, do you have exudate, mucus production, abnormal lesions? And all these results from these different tests here are going to be placed into this this chart. On this paper here, you can see the source of the specimen. Okay, what test is requested to be done? Let me put my little thing here. So, source of your specimen, the test that is requested. Do you want a gram stain? Okay. What are the results of the culture? Is it anaerobic, aerobic, yeast, other things? All right. Is it a parasite or fungi? Okay. All these different things for this test. Everything below here is what the lab is going to do. Okay. Antibiotic susceptibility testing down here can be requested or performed. Right, so direct microscopic observation of fresh or stained specimens is one of the quickest methods to determine presumptive and then also sometimes confirm microbial characteristics. Okay, it's really important to help determine cell size, shape, and arrangements of cells, which really helps you to identify. The most common two types are going to be the gram stain and the acid-fast stain. Okay, so here on the left, and you should be familiar with this in lab, is a gram stain. You've got both gram-positive rods, okay, so bacillus, and gram-negative rods. So that's the pink rods here, that's bacillus, and the gram-positive cocci. So this is the round berry shaped here. So you've got two different things here. All right. And so then you have this acid fast or differential stain where you've got red purple cells that are acid fast and the blue cells are not going to be acid fast. Trying to get rid of this little light here so I can stop the recording. So here we have selective and differential growth medium. So I've got three different types here. We've got specialized media, which is going to be used to enrich a pathogen that is present only in very small numbers or can overgrow very easily. We have selective media, which is used for non-sterile specimens to encourage the growth of only the pathogen that we are thinking is there. And then differential media can help us to differentiate basically between two different species that may be similar. All right. So non-sterile specimens like urine, fecal specimens are going to require both incubation in differential media and selective media. So we'll often use these in conjunction with one another. So UTIs, urinary tract infections, are often caused by what? E. coli, right? That's a pretty nasty one. So selective media can be used that will be sure to allow the growth of that particular common pathogen. Specimens are going to be inoculated into differential media to identify defining characteristics in blood agar, or fermentation of sugars like McConkey agar. All right, so here's some examples. Here we have plain old regular nutrient agar. We've got E. coli, Staph aureus. Can you tell any difference? Not really, okay? So that's not really going to be helpful for us. Now, we could pull it off and do a gram stain, but we can also put it and grow it also on this particular McConkey agar here, which will be more selective. It is not going to allow the Staph aureus to grow, but it will allow E. coli to go, okay? And it's red because this is also going to have blood in it. This is blood agar, all right? And so over here, where that would come into play would be where you have E. coli, which does not do the hemolysis here compared to salmonella, which does, okay? A particular serotype of salmonella. The cells here, it's breaking down, that clearish area is where it's breaking down red blood cells, okay? And you can also look to see, are they fermenting a particular type of sugar, carbohydrates, that type of thing. So this is showing you a dichotomous key, which is basically a flow chart that helps you to identify specimens. And so we're going to look here at gram staining, growth on differential media, and then just some simple enzymatic tests to kind of narrow down the field of what we're looking at. So here, you have a cockeye, you have berry. So you're going to gram stain this and find out, is this gram positive or gram negative? So you've looked, you see that it is round, or you may not even look, you may just go ahead and start with a gram stain, okay? So if it's gram positive, then there's a series of tests you may want to do. You want to know, is it catalase positive? Does it use catalase enzymatically, or is it catalase negative, okay? And then it's also saying you're going to have these particular species are generally going to be tetrads or irregular clusters. These are generally catalase negative, is usually going to be streptococcus, okay? And this is a very narrow range here, not 100% inclusive of all species, okay? So if it's catalase positive and you have irregular clusters or tetrads, you want to know, is it anaerobic or aerobic? Because you want to differentiate between micrococcus, which is actually in some areas normal microbiota, and Staphylococcus, okay? Some Staphylococcus species. This one is Planococcus species as well. And so if it is slightly aerobic, you will put it in here into the tube, and if you see bubbles occurring, you know that it is fermenting, okay? And sometimes it's a color change. It really just depends on the particular test, okay? And what it's producing. Is it producing a gas? Is it using this particular sugar, this fermentation? It may cause a color change due to pH, okay? It depends on the test. So gram negative over here, then you want to see, is it aerobic? Is it anaerobic? Is it oxidase and catalase positive or negative? So several different things here. And so that can be broken down then further by these enzymatic tests on down here into these, is it Neisseria or Avianella, okay? These other species here as well. Well, trying to get this off. There we go. So these physiological reactions that we were talking about of bacteria to nutrients, like if they break down catalase or oxidase, or do they ferment certain types of sugars, other substrates, give us a lot of evidence of enzyme systems in certain species. So if we know which enzyme to isolate, it can help us identify. So many tests are based on these enzyme-mediated reactions that will cause a color change in certain medias, okay? And so it's going to be based on those reactions and visualized then by a color change, which is a pretty easy diagnostic tool, okay? And like I said, sometimes you'll also have, especially with fermentation, whether or not they produce a gas. So a microbe is cultured in a medium with a special substrate and then tested for a particular end product, all right? Microbial expression of the enzyme is going to be made visible by that colored dye, so or pH, response to pH, one of the two. So no coloration then would mean it lacks the enzyme to utilize that particular substrate. So biochemical tests include things like testing for catalase, urease, oxidase, the different sugar fermentation. Does it ferment mannitol, mannitose, glucose, sucrose, those types of things? And then, like I said, can it produce a gas? All right, so here is a rapid biochemical test. You can see this is kind of a microarray type test where you have a well plate with a bunch of small little wells in it. And then you're going to have these different tests here. So before inoculation at the top, each small tube in the strip here, it's going to be very small, contains a substrate like a carbohydrate or another substrate for an enzyme that the unknown microbe may have, and that's what, like ARA, may be arabinose. Okay, let's see. So samples of a single bacterial isolate, which can be like an unknown gram-negative microbe, can be placed in these 20 different little cups, okay, or tubes, which the growth media and chemicals are designed to test for those individual enzymes, like arabinose. So the organism here is going to produce positive results. In this case, it's going to be 4 positive results here. We've got LDC, ODC, IND, or GLU, which could probably represent glucose and some other substrates that a particular enzyme may act on. So after inoculation with the unknown sample, the colors of the wells can indicate that positive or negative reaction. And the particular combination of colors are actually converted to a code which indicate the identity of the bacteria because it's going to be referenced against a database. All right. Here is another phenotypic test. This is the MGIT, Mycobacterial Growth Indicator Tube. Fancy name there. This is going to detect the growth of the slow-growing Mycobacterium tuberculosis, all right? And so this is going to monitor the oxygen levels in a tube that's inoculated with the patient's sputum or blood. And then there's a silicone chip at the bottom of the tube that is actually impregnated with a fluorescent substance that is sensitive to oxygen levels. And it will actually fluoresce as oxygen levels in the medium decrease. And so automated machines then will give a visual and audible signal so a positive culture can be easily identified. So almost as important as identifying the pathogen, we need to figure out which medication we want to use. Which one is going to be most effective? Are we dealing with antibiotic susceptibility? So since we have that particular issue and other issues, this is really useful in determining which drugs can be used in treatment. So with the crisis level rise in antimicrobial resistance, it's really important to not waste valuable treatment time by guessing about which drugs to prescribe. Most automated phenotypic systems are going to include this during pathogen identification. This is also useful for species of Streptococcus, for Pseudomonas, and Clostridium species. So this is another type of just kind of a miscellaneous test here. This is for phage testing, like bacteriophage. So this technique involves inoculating a lawn of cells here, of bacterial cells, on a petri dish and mapping it off into blocks, and then applying a different particular bacteriophage to each section of area of growth, again, to see which is going to be effective. Cleared areas are corresponding to lysed cells, so that indicates the sensitivity to that particular phage. Bacterial identification can be determined based on using this particular unique pattern. We also have issues with things like we need animals to cultivate Mycobacterium leprae and Treponema. We have avian embryos and cell cultures that we use to grow host cell-dependent things like Rickettsia rickettsii, chlamydia, and other viruses. So there's a random rate, not random, but there's a range of miscellaneous tests out there that we can use. All right, so what is the clinical significance of our culture? It's important to quickly determine if something from a specimen is important or is it actually just normal microbiota. Hence the reason we need to learn what the normal microbiota of a system are, which is what we're about to learn in with the next 6 chapters. What is normal microbiota is important, right? Because you don't know what's abnormal if you don't know what's normal, and you're usually not going to have one particular microbe in a culture. You're going to have several because you're going to have all that normal microbiota. So you need to know, is this contamination? Is it normal microbiota, or is this a pathogen? Okay, so a few colonies of E. coli in urine can indicate normal microbiota, okay, especially if it's not clean catch, okay. Several hundred obviously is going to be indicative of an infection. A single colony of something like Mycobacterium tuberculosis in a sputum culture or an opportunistic pathogen in a sterile site is highly suggestive of the disease. So it depends on what you're looking at, all right? And so then you would need to isolate a pure culture of any microorganism to be sure if it is actually the agent causing the disease. Some of the drawbacks of phenotypic methods are when the microbe has to be cultured, it takes at least 18 to 24 hours, usually longer, to get a nice lawn of bacteria growth there. Many infectious conditions also may be caused by organisms that we can't culture. And so that opens up the possibility that the organism that we do culture is simply just a bystander, just happens to be there. So looking at the direct examination methods here for phenotypic still, microscopy of patient specimens, usually after we stain, don't have to then be cultivated. Other methods require cultivation. Selective or differential growth. Use of specialized media that is going to identify characteristics like colony appearance, whether or not they are motile, do they produce a gas, those are important, okay? Those require cultivating. Biochemical testing, the growth of the microbe in the media is going to detect the presence of the microbe's enzymes and create basically that metabolic fingerprint, okay? Susceptibility testing, if there's a particular pattern of susceptibilities, this can help us to identify the microbe. And then there we have our last 3, our little miscellaneous. So that kind of sums up the phenotypic methods that we've looked at. All right, so finishing up the first half of chapter 15, let's do a little NCLEX prep question here. So when determining the clinical significance of cultures, what do we need to consider? A, the number of microbes is important. B, the presence of a single colony of a true pathogen may indicate the presence of the disease if the culture comes from a site, I should say site, known to be sterile. For example, the cerebrospinal fluid. So the next one is the repeated isolation of a relatively pure culture of any microorganism can mean it is an agent of disease, although that is not always the case. D, a range of tests may be needed to identify a pathogen, or E, all of the above. So pick your answer. The actual correct answer here is E, all of the above. All of this is true and relevant.